think it's recording. Yeah. Okay. So basically, your primary protein chain your primary structure is just gonna be a long polypeptide chain. And then we move into secondary. And secondary is when they're called alpha helices and beta sheets. So alpha helices, they kind of look like little like spins like this throughout your protein structure. Beta sheets, on the other hand, look something like this. And I can pull up like what a protein looks like as well to kind of get a better view of protein. So it kind of looks something like this. So see these dots? Those are your amino acids, and they create this helix within the protein. And when we're looking at like a crystal structure, it's going to look something like this within your protein. And then beta sheets start looking like this. This is what a beta sheet looks like. And one thing I want you guys to note is that in your secondary structure, the bonds between your amino acids are between your amine group and your carbonyl, which is part of your carboxyl group. So I think that's one thing that I want you guys to write down. So
Can
the
you explain that?
Yeah, so do you remember how I talked about hydrogen bonding last week? How like water is like a prime example of hydrogen bonding?
Yeah.
Like this. And so we have these like temporary bonds that form between our oxygen and our hydrogens. And so when we're looking at like a secondary structure, we're only looking at the hydrogen bonding between the amine group and the carbonyl on the amino acid. This is not involving our R group. This is not involving the hydrogen off of our main carbon. It's only between this oxygen and this hydrogen that it's forming these hydrogen bonds to create either beta sheets or alpha helices. Give me one sec.
How are you guys? Good, how are you? Good. Wait, it's amine and what's the other one? Carbonyl. Carbonyl.
Okay, so this is our like technically you'll have like your OH group off the end of this. So this is our carbonyl and all together this is our carboxyl.
Wait, are you like showing something right now?
Oh, is it not sharing my screen? Oh, it's not sharing. One sec.
Yeah, YouTube. Yeah.
Okay.
Yeah,
Here. Do you guys see that?
oh, okay. Oh, okay. What's your face? Oh, tea or coffee, let me know. Thank you. I got apples, that's it.
Can you guys still see the screen? Or is it
Yeah.
not?
Yes.
Okay.
We can now.
Okay. So these are between amine, amino and carbonyl oxygens and hydrogens. And so this is all hydrogen bonding. Do you have that down? Okay. And then we move into I'm going to bring the tertiary down here. So tertiary is when we start having um, reactions or interactions, intermolecular interactions between our R group. So R group interactions. And so a good example of this would be, you guys know how you learned about, like, I think your sulfohydryl group, which kind of looks like this.
Mhm.
So this is the amino acid that has this sulfur group is called cysteine. And cysteine, for example, can make a bridge between two cysteine amino acids. So let's see. It's called a sulfur sulfide bridge protein or the disulfide bridge. And so it kind of looks like something like this. So we can have like an interaction like this. See how it's coming from like a cysteine to another cysteine? So that's like a type of bonding you can have. Um, you can also have hydrogen bonding if if like say an amino acid has like an OH group, we can have some bonding between these like this. You can have like ionic or like when we have like a say remember how I was talking about like acids and bases and how proteins can sometimes lose their hydrogen or they can gain a hydrogen? So, depending on their base charge at like physiological pH, which is like around 7, these two can attract. And so you can look at that. Ionic bonding. You can also have it with like your like if an amino acid has like an amine group, we can have some of these like partial positive and negatives um interact. So here we would have like I also talked about this last week, how nitrogen, if it has 4 bonds, it has a net positive charge because it only likes to have 3. And then if oxygen only has 1 bond, like this, and here are the lone pairs.
Can't
Um,
see it again.
oh, my bad. Like this. How the amine group, if it has 4 bonds, it's positive because it only likes to have 3. And if the oxygen is like, it typically likes to have 2. That's why H2O is very like a neutral molecule. If I added like a hydrogen here, the net charge would be positive. But if I lose a bond, it becomes negative. And so we can have an attraction between these charges. And so that would be like an ionic bonding in proteins. And then, so say we go through like all of that. We then get to, it's called quaternary. And so something really interesting about proteins is they can have multiple chains within a protein. So we can have like multiple polypeptide chains within a single protein. So say I have something like this, say this is like what my protein looks like, which is they quite literally look like that. And I can have like another chain in here going like that. I could have like another one like this, or, another one like this. So I could have like, so this would be like four chains in a protein. And so this is where we move into like quaternary is when we have like interactions. We can have R group interactions, and we can also have just like, um, remember how we were talking about amino acids can be hydrophobic and like hydrophilic, or they can have, yeah, hydrophobic and hydrophilic when we have something that's polar and something that's non-polar. So polar, remember, is hydrophilic, and if we have something that's non-polar, it's like oil. So it's not going to dissolve in water. It's not going to like water. So that's when we get our hydrophobic. And so when we have proteins like in our body, remember how we're mostly made out of water, or just like a cellular environment is going to be polar. Our polar amino acid R groups are gonna like start facing. So if we have like a bunch of water out here like this, and I'm gonna draw like this protein as some, it's just like a circle. So say this is our protein. My polar amino acids are going to start facing outwards because they like water. They want to be near water. They can do some hydrogen bonding, they can have some weak interactions, but overall, they want to be near the outside. And my hydrophilic or my hydrophobic amino acids are going to start going in towards the middle because they don't like water. They don't want to be there. So they're going to go towards the inside. And we will have hydrophobic, and this is also known as nonpolar. Does not like water. So it's going to be something like that. And that's how these like R groups and like these tertiary groups or tertiary structures are going to start to form. And within something like this, the chains will also have interactions between them. So like say we have like two hydrophobic, or say we have some like OH bonding or hydrogen bonding are, so this is chain one and this is chain two. And so we're going to have some like weak interact hydrogen bonding interactions between them, and this is going to kind of be how the chains start to fold together into a protein, if that makes sense. And so what I would know for these is just main chain, amino. I'm just going to write this as carbox, H bonding, R group, and interactions between chains. And so that's kind of what I would know for proteins. I don't think you have to go as in depth, but sometimes I think like being a little more in depth with what you know can help you work through certain problems. So that's what I would know for this. Um, yeah, do you guys have any other questions on like this part?
I think so.
Okay. I'm just trying to make this so I can see you guys at the same time. think that's good. Okay. So now that you guys have proteins and you kind of know some of those interactions, should we move? Okay, so we got these two done. Should we move on to carbs? Are you guys good with moving on to carbs?
Yeah.
Okay. Let me find, I want to find a picture of,
Yeah, there's a fountain here, but what's that? I want to go under the. I'll show you.
okay, so let's see. Have you guys looked at, so this is called a Fisher projection. Have you guys looked at these or has it just been this?
Kind of both, but
Kind of
I don't
both.
officially thing.
Okay, actually, I think this one might be a good picture to look at.
I don't
So,
know fishing.
what's that?
I don't what the fishing thing? What'd you say?
Oh, okay, so this this one right here, this is called a Fisher projection of a carb.
Because it
And
looks like a
it's
fish.
I guess if you want to think of it like that, it's spelled like this. And this is perfect. So we can look at these too. And so basically what happens in this type of like interaction, so this turns into this. And basic so it's just turning into like a cyclic molecule. Cyclic is like anything that, can be like anything like this. This is cyclic. This can be cyclic. This. These are all types of molecules you can have. So this is like you'd say cyclo, and we have one, two, three, four, five, six. So it'd be hex. And then because we have all sigma bonds, or we don't have any like double or triple bonds, we get ane, as in alkane. So we know that it's a cyclic molecule with 6 carbons and it has alkanes. If does that make sense?
What's an alkane again?
That's just like single bonds like this. This is alkene. This would be like an alkene. Or it can literally just be like this. And I have 2 carbons right here, like that. I think you guys talk about these in lipids as like unsaturated and like saturated. I know has been brought up.
Can you move your
Um,
voice so we can't see that stuff?
oh, there. Can you see it now?
Yeah, thank you.
Okay.
What was your question?
Um, Give me one sec. I'm just trying to hide the meeting controls so I can see. Okay. Um, so does that make sense for how we get something cyclic?
Yeah.
Okay, for naming. So basically what happens is that um we have this oxygen right here will attack this bond right here, and then we'll get something that looks like this. So I'm gonna just draw this out so I can show you guys like a written visual.
Wait, so does it become like a little acidic, or what do you mean, like a tap?
Um, it's like hold on. Give me one sec. I'm just writing down the what the sugar looks like in the Fisher projection, and then I'm going to show you. I'll switch to the other tab, and I'll show you what that turns into. Okay, do you guys see this right here? So remember how I said like oxygen is pretty like electronegative?
Mhm.
So we kind of have like a partial negative right here, a partial negative right here, and there's kind of like a partial positive right there. So what happens is that this oxygen right here is very attracted to this partial positive, and that's when we get this structure. So I'm gonna also write this down. Okay. So do you guys see this now? So I'm gonna highlight. So this carbon right here and this right here will be these two carbons right here. Does that make sense? It's kind of like wrapping around.
Oh, okay, yeah. Wait.
And like this one right here gets turned into like an OH, which I think is right here. And this is right here. So does that make sense between those?
Hmm.
Okay. Um, and something else I want to go over is like monosaccharides. So this is like glucose and this is galactose, and basically they're like almost the same thing. The only difference that we have is this right here. See how they're like flipped? Or I guess we have one right here too. Glucose. And so the differences between these like six atom rings is the conformation, which is what you call it when you have like an OH or H and they like these flip positions. They give us different conformations with different properties, if that makes sense.
So how does flipping one thing change it? Like, why does that
Um,
happen?
I guess it's just like I'm not sure like the exact reason, but I guess it probably has something to do with like metabolism and just how these like molecules are formed. But it just like remember when you guys were tasting like different sugars in
Yeah.
class? Um, did you guys try like galactose and glucose?
Uh-huh.
Yeah.
Yeah, so like that would be like the difference. Like they taste different. And so basically, like these molecules, or like this center right here on each of these, are considered chiral because they have like chiral's kind of a big word that you guys probably don't know yet, but the chirality of a carbon can determine its like optical properties. Do you guys know like polarized lenses? Like, do you know what those are? Like for glasses?
Kinda.
Like, you know how it can like help you see into like if you're fishing, it can like help you see the water without the like glare on top, like that reflection.
Yeah. Yeah.
So basically what it's doing is it changes it rotates the light. And I'm not going to get like super into it, but it can completely change like the difference of how a molecule smells, how it tastes, just like other like physical properties. And so that's why these are starting to taste different, is because of those properties. Like fructose, that's one thing you should know. This is just like it's considered like a pentose. This is a hexose because this ose right here basically means like sugar. That's kind of like the giveaway. And if you see anything like this, like see how we have like this oxygen in the ring? That's going to be like a dead giveaway for a carbohydrate. Same with these. If you see something like this, it's probably going to be a carb. If you have this C2OH on the end and this like right here, that is going to be a sugar or carbohydrate. And then if we get into like disaccharides, have you guys done those yet?
A little bit.
Basically, it's just the link of two sugars like this. So like here's a good one. Um, and this might be something you want to write down as like examples of disaccharides. I don't expect you to know the structures, but sometimes it's helpful. So like sucrose is composed of glucose and fructose. So remember how we went over this as a hexose and this is a pentose? They basically just stick them together and that's how we make sucrose. Same thing with lactose. We have one right here, which is our galactose, and then we have a glucose. So know that like lactose makes, or galactose and glucose make lactose, or glucose and glucose make maltose. Those are some pretty like common disaccharide examples. And did you guys get into like why these are called like beta or alpha?
No.
No? Okay. Then I won't worry about it unless you guys are dying to know.
I'm good.
Okay. So then next we'll go. Are you guys good with carbs now? Does that make sense?
Yeah.
Okay. So let's go to a lipid structure. You guys went over like triglycerides, right?
A little bit.
Okay.
Maybe a little bit.
I've heard the word.
Okay.
That
So
was one.
Oh, the king.
It was the jellyfish.
It is the king.
yes. So remember how I was talking about like alkanes and alkenes?
Yeah.
Yeah. So I'm gonna zoom in right here. So if I have like a, say, here's like the lipid head, and this is the body. Saturated means it has lots of carbons on, or lots of hydrogens on it. It's saturated. And then when I have something with that kink, that's when we're gonna get a double bond and it becomes, so this is saturated and this is unsaturated. And that's how you can tell the difference between the two. Does that make sense?
But so that double bond just makes it unsaturated.
Yeah, because we go from two hydrogens on each to one.
Use. Okay. That's cool.
Oh, is that the one that's like alternating directions? And that one can't. Yeah, it's spin. Yeah, and it's like less of them can like pack into a space or something like that.
So that's like about it. When saturated is like
It's not. Yeah. Wait, which one would be like butter?
It's like the cis orientation, the trans
Yeah, yeah,
orientation.
yeah.
And then there's empathetic.
Oh, yes, this is the same side trans as ours.
Yeah, like
Yeah,
so just
so
the double
like
one.
this would be considered cis because
Mhm.
I have my two hydrogens here. The one that I drew out would be this would be trans. So basically the difference between a cis and trans alkene. So I'm gonna give you just like an example of just something pretty like simple like this. Say I have like an OH here and an OH here. So these alcohols are considered like higher priority groups. There's kind of like a rank list of them, but I don't think you need to know that. But we know that there's also a hydrogen right here. So we have one, two, three, four bonds coming off this carbon and this carbon. So when we are looking at the end to determine if something is cis or trans, we know that we have a higher priority group here and a higher priority group here. And when the two higher priority groups are on different sides, that means it's trans. If I have something like this, I have an OH here and an OH here, this would be considered cis because my two higher priority groups are on the same side.
Does it matter if like the groups on the side are like different? Like, because
Um,
right both OH doesn't
So
matter if it's
like
like
say if I had like something like this and I have like an OH here, like an SH here, it's still going to be higher priority than H. But what if I have a carbon here? There's a or yeah, what if I have like a carbon right here? CH3. Would OH or CH3 be a higher priority?
CH3.
Not quite.
how do we know
Oh,
something's high priority?
so OH is going to be the higher priority just because usually things that are a little more electronegative are going to have a higher priority. So like oxygen over
Not quite.
nitrogen, stuff like that. And so I don't think you have to worry about that too much. Just know that like see in my unsaturated right here. So I'd say this is trans because I'm choosing between a hydrogen and a carbon right here, a hydrogen and a carbon right here. And we know that carbon is going to be a higher priority than hydrogen. So I have one going this way and I have one going this way. So this is going to be trans. And then if I had something like this, which would make that kink, would be I have a higher priority here, here compared to my hydrogens coming off the carbons. So this would be cis trans. Again, I don't think you have to know like the fine details, but like this right here would be considered cis because we have our hydrogens, which are our lower priorities, and our carbons, which are higher priorities than hydrogen on the same side, like that.
Oh, okay.
And so that's how it kind of like makes that kink. So if we move into something like this, see if I can zoom in good. Okay, so you guys went over like the lipid bilayer, right?
Yeah.
Yes.
Yeah.
Okay, so kind of what makes this is we have a phospholipid molecule and we have fatty acid tails. So these fatty acid tails right here, these are going to be the hydrophobic. So they're pointing inwards. They don't want to like touch the polar environment of the cell. And then we have the hydrophilic head, which is going to be a lot more prone to just like being in a polar environment. It likes it. And that's because um we have this like phosphate group right here, this phosphate head. And so technically, this is going to be a neutral molecule just because we have this N plus here and this O minus right here, and they cancel each other out. So they're going to be pretty neutral. And then this is right here. This is a fatty acid. So see how it has this really long carbon tail right here? That's going to make this molecule pretty hydrophobic, even though we have this right here. Even though we have this carboxylate group here, this is going to be pretty hydrophobic. And so that's how we get this phospholipid bilayer right here. And so basically, if you see anything with this like really long tail like this and kind of a polar head, that's going to be a dead giveaway for a lipid. Does that make sense?
Mhm.
Okay. I know you guys looked at triglycerides. Okay. So right here, this is a triglyceride, and we do have some like unsaturation within it. It doesn't really matter. All I need you guys to know is that we have a glycerol, a triglycerol backbone right here. And then they connect with a fatty acid right here. So if I go back here to explain everything, a glycerol backbone basically just looks like this. So that is what we consider like a glycerol backbone. See how I have my carbons right here? And I have like my oxygens right here. Um, Here, this is perfect. I like this. Can you guys or let me click on it and then I can zoom into it. So can you guys see this pretty well?
Mhm.
Okay. So this is my glycerol backbone right here, right? So I have one, two, three. Triglycerol. And that's kind of where it comes into this triglyceride. So I have three fatty acid chains, and these interact with these right here, these oxygen and hydrogens, my OHs interact, and I form this triglyceride right here. And one of my byproducts is water because I'm taking out these hydrogens and oxygens right here to make H2O. That's why it's a byproduct. So, looking at what you guys have done with creating bonds, we have your like hydrolysis or your dehydration synthesis. Which one do you guys think this would fit more into?
Dehydration synthesis.
Yes, because we're creating a bond and we have H2O as a byproduct.
Yeah.
Does that make sense?
Yeah.
Okay.
Here we go.
It's clicking. That's good. We want it to click. See, and then here's like another good picture of that. We have our glycerol right here, our fatty acid chain. Okay, so I think we've basically gone through all of these. Carbs done, lipids done. Okay, let's get into our double helix and then DNA/RNA. We did this. So let's move that down here. Uh, Actually, we'll go up here. So, do you guys know what these stand for?
Deoxyribose. No, what? No, I don't.
Deoxy Ribo. Here, I'm gonna actually pull up the DNA structure.
Oh, deoxyribose nucleic acid.
There you go.
Like that's right.
So
Hey, I did the hard
I know. No, I got the end of it, so I'm feeling that this is team effort.
I think we also kind of went over this um last week a little, but I can explain again. Deoxyribose. Nucleic acids. So these are all nucleic acids. And then we just have. So do you guys know what do we have in a nucleic acid?
Wait.
Like what makes up a nucleic acid?
Nitrogen.
Topic one.
What like specific macromolecules and like functional groups?
Oh, look.
Like carbon.
Hold on.
Back to this. So if you guys look at this, what functional groups do I have here?
Carbonal.
Yeah. Um,
Mhm.
maybe in the bases, but I have a base. What macromolecule is this?
Oh, that's one of them.
Is that a lipid? Is that a carb?
Uh, I
Like all carbs.
what? Oh wait, no, it has the one oxygen thing. Is it? Is that
Yeah.
not a
Carb.
is
Yes.
that not
So we
is
have
that right?
we have a phosphate right here. We have a carb right here, and we have, see these bases right here?
Yeah.
These kind of like your like amino acids, you know how you have like that variable group? They belong right here on this base. So, you know how we have like this right here? I'm gonna actually draw this. So, I'm gonna write P for phosphate and um, so basically what we have is the sugar phosphate backbone of DNA. And off these carbs, I have my bases, right?
Mm-hmm.
And as we can see in this, this is what these, so what are some functional groups in here, in these bases that you guys see?
Carbonyl.
Yeah, we have a carbonyl. What about with the like nitrogens?
Amine.
Yeah, we have some amine groups. Um, yeah, that's kind of all you have to know for that. We have some alkenes in here. Like this is an alkene. It's an alkene. This is a methyl group
You know.
How do you know it's an alkene?
because it has double bond and it's only
Oh,
carbon.
okay.
So an alkene is just like a double bond between two carbons. An alkyne is a triple bond between two carbons, which I don't think you're gonna run into many of those, but they exist. Um, yeah. And so do you guys see right here how we have one base, which is adenine, and we have a thymine right here? See how they're hydrogen bonding with each other?
Mhm.
This is how we link DNA. So these links right here is this. This is what's holding these two strands together. So if I go back here, I have a base, which hydrogen bonds, which I'm actually going to write like this. Hydrogen bonds with another base. And then this is connected to a carb phosphate. I'm just going to write C in here. It's not carbon, carb, C, and a base right there. So imagine if I do a bunch of these. I'm going to start getting this ladder, right? Like that. So that's how this ladder is being formed in DNA. Same thing with RNA, except instead of thymine, we have uracil. So that's like that's the main difference. They have the same structure. It's just we have uracil instead of uracil, not thymine. Does that make sense?
Yeah.
Okay. And what I want to go over. So you guys are doing the five. I want to find a good picture. So like here's another kind of what this looks like. Phosphate, sugar, and then your base. And these are our bases. Oh, one thing to know is that G and C, when they bind like this, they have three hydrogen bonds, which means they're stronger. So DNA with more CG content is more like resistant to denaturation, which is basically when it just for something to denature, it just breaks apart. So like we don't want DNA to break apart. That would be really bad. So that's what these hydrogens are for. It's like pretty strong bonding. And that's what holds your double helix together. And I think you guys will get into like single-stranded stuff further into like transcription and like translation. I don't think you have to worry about that now. And then I want to see 5' and 3'. Okay, so see this right here. I think this is actually a really good picture. So when we're looking at DNA, we add in the 3' direction. So I think this is one of the things I know you guys wanted to cover. So we add in 3' direction, and prime is basically it's labeling what carbon we're looking at. So add in 3' direction. So when we build DNA, we're going from the 5' end to the 3' end. And one thing to know about DNA is that it's anti-parallel, which means it's like this, but we have a 5' right here and a 3' right here. So this is what DNA looks like. And this is mostly for like replication and transcription processes. And so when we're adding to like not our bases, we're not talking about our bases, we're talking about the sugar phosphate backbone. So this thing right here. When we add to it, we have a carb, right? Like this. We have our base. We have an OH group right here. And we have our phosphate right here. which I'm just gonna write P. And so basically, what we want to do is we want to label our carbon starting from right here and going this way.
Thank you for going to
Can you
switch tabs
can you
again.
show?
Oh, my bad. Like this.
Oh, shit. I got it all.
Oh,
And so when we add, so here's my 5' carbon right here. Here's my 3' carbon. So when we're adding, we want to add to this side. And do you guys know why we can do that?
no.
Because it's an OH. Because like the OH can go with the.
Yeah, we can very easily take this hydrogen off of it to create a bond with another phosphate. So this 5 will have a 5' end come in and bond with this OH, right? Like this.
So
And
like where
see
that O
how,
is.
yeah, see this O right here? That's representing this right here. Let's see if I can find a chain. Here we go. Can you guys see that?
Yeah.
So see how I have my 3' right here? My 5' is right here. This is my phosphate. So they call this phosphate the 5' end just because it's coming off the 5' carbon. So 1, 2, 3, 4, 5. One, two, three, four, 5. So here's my phosphate, and it's binding with that 3' OH or oxygen. We just don't have the hydrogen anymore. And this is through your dehydration synthesis or your condensation. That's how we're creating this bond right here. Does that make sense?
Yeah.
Okay. Do you guys have any questions on DNA?
I
So
don't think so.
Okay. Um, okay. Yeah, RNA is basically the same thing. I can show you a
Wait, DNA or RNA? RNA.
Oh, I meant to say RNA. Oops.
It's like wait, you
Yeah.
added a
So
new.
it looks like this.
So does RNA only have like one swirly side?
Yeah, RNA just will have one strand. And it's because each strand of DNA can make one strand of RNA. So we'll get like two separate strands.
Yeah. Yeah.
So like this. Say like this strand right here will make this strand right here. If that makes sense. And see, the only difference right here is we have uracil instead of thymine. Oh, and like this sugar right here. So DNA, deoxyribonucleic ribose. So I think that's why I wrote it somewhere in here. But that's where your sugar is coming from, right here. And then we wanted to go over the Hardy-Weinberg, right? Last like couple of minutes.
We need to use Hardy Weinberg for this unit or no?
Yeah.
Just a refresher.
Oh, just that.
Mhm.
We don't have to.
I get like the concept of it, but I don't know how to like use it.
How do yeah, I know,
Yeah, like I'm good on like what it is. It's just the like application of it. Mhm. I don't think
Yeah.
anyone understands.
Let me look up an example really quick. No, look up.
Marla, do you think that's accurate? No, like we all like know what the equation is and we know like what it is. We just don't
But just
know.
like how to use it.
Like
yeah.
like some of the questions would say like frequency, but there's another equation where it's just like p plus q equals 1.
Yeah.
And then you'd have to figure out like what's the percentage versus the frequency.
Yeah.
Like,
Okay.
but I can pull like the AP test questions. No. Like from our test. Do we want to do that or no?
We could, I think
Do our examples.
IDK. Or IDC.
Identical.
They don't know anything. Wait, if you hit on the Zoom thing, if you hit like one of the nine dots, does that show every single person? I think for another 5 minutes. Is this still 4:30, Mia?
Um, yeah. I mean, but we can like if we want to do one of these problems, we can do that too. It's not that big of a deal.
Okay. Okay, chill out.
Okay. I think I have one of the problems from one of your packets.
Okay.
Um, so let's say we have a total of 700 cats.
I'm like eating, please.
I actually write this up here.
I'm still gonna get anything really. I'm just tripping over. You were okay. You did. You did. Yes, you did.
So, and we're gonna have our dominant allele equals 0.81 and our heterozygous is gonna equal 0.16. And we want to know what BB equals. So we want to know how many cats have the how many have this.
Let's just add them and subtract that one.
Yeah, so what we'll do is we'll do 0. So BB plus big B plus little b is going to be equal to 1. And so we'll add these up. 1, 6 plus BB equals 1. And then we can find out what that equals. And I think it's point 0.303 equals
Mhm.
BB if you do the math on that. And so this is a percentage. This is a percentage. Or we can turn it into a percentage. So technically, this would be like if we multiplied this by 100, this would be like 3%. 3% of the total population are BB. And we know we have a total population of 700. So what could I do to find out the number of cats with
Multiply,
BB?
multiply by 0.03.
Yeah.
We would get equations too, though, where like we didn't know big B, little b. We would only
Yeah.
have BB and we'd be asked to find
Okay.
what
Let's
percent of
do
the population was little b, little b.
Okay. Let's do another one. So that's like how you would do that. And technically it like represents Hardy-Weinberg, but we can do P equals 0.45. So a population in Hardy-Weinberg equilibrium has P equals 0.45. What percentage of the population is homozygous? So do you guys know what we would do for our first step?
We'd find Q.
Yeah, we can find P or Q. We can do that. So what we're gonna do, or let's find, well, we're gonna set this as P first. Because we know what P squared plus 2PQ plus Q squared equals 1 in Hardy-Weinberg equilibrium. So what we can do, let's see. Yeah, let's find Q. So we know that, let's see. P plus Q equal 1. That's like another thing that you should know. P plus Q equals 1. So we can find Q by putting in 0.45 plus Q equals 1. How can we go from here to find Q? Yep, we can subtract, and that will give us 0.55 for Q. And so now we can calculate the frequency of each by, let's see. Oh, and also one thing to note, this is um, homodominant, and this is going to be our homo recessive because this one will be like big B, and this will be little b, little b. And so we want to find this. And so now we can actually calculate what P actually equals. So P squared equals 0, or yeah, 0.45 squared. So that will give us our P squared, whatever P squared is equal to, and we'll be able to plug that into here. So I think that gives us,
Wouldn't you square root it though?
no, because if we have P equals 0.45, we want to know what P squared is.
Okay.
So we would have to square our P value. Does that make sense?
Mhm.
Okay. So I think that gives us, I have this like written down, 2025. So that is what our P squared is equal to. And then now we also have Q, so what else can we find?
You squared.
Yeah. So Q equals 0.55 Q squared, so we're going to square this, is equal to 0.3025. So that is what Q squared is equal to. And let's see. Okay, so I think we wanted to, okay, we want to find the number, the percentage of the population that is homozygous. I think I forgot to mention that. Yeah, what percentage of the population is homozygous? So now we have two frequencies here. Or we have now we have percentages. These are our percentages. And these were our frequencies. So that's one thing to note. Frequency and percentages are different. So now what can we do to find the percentage of the population that is homozygous?
Add them together.
Yeah. And so when we add these percentages together, we get 0.505. And then when we multiply that by 100 to, put it into the percentage, we get 50.5% are homozygous. Does that make sense?
Yeah.
Okay. So that'd be like an example of putting it in. When you're like dealing with like frequency and then like percentages, because these two are different. Let's see. Let's see. I want to see how I can make this easier. The frequency is just going to be P and Q, like just your plain values, not squared. So if you're given frequency, you can set them equal to P or set it equal to Q if you're given frequency. And then if we're given percentages, this is when you're going to have like your P squared and Q squared. So if we want to find what P is equal to, and we're given P squared, so if we're given like our percentage in a decimal, we'll just square root this. And this will give us P, which is our frequency. Same thing with Q. You do the same thing with Q. And then you can figure out what 2PQ is too, if they're asking. Do you guys have any questions on that?
Oh,
That kind of clear that up.
yeah.
Okay. I'm sure there's like, if you guys are like really struggling with that, I would just like look up a video and, take like 10 minutes to do it.
That's not working.
Keep talking.
Thumbs up. Please.
Marla, get you to you're in camera.
Thumbs up.
Oh, Charlie, you're in camera. Oh, it's me. Thumbs up. And I can do this.
Or you have to hold it.
Marlo, thumbs up.
Wait, keep it. Wait for it.
Let's get it overstimulated.